maybe it's because I didn't want anyone else to know who I was. All right. So, the title of my talk is Bacterial Source Tracking, a Toolbox Approach, and what I'm going to do is describe basically some methodology developments that we have had for several of our bacterial source tracking projects over the last 5 or so years. So basically what we started out with for selection of methodology, what we determined from some of our previous studies, some of our current approaches, as well as the recommendations that were given to the state of Texas by the Texas Bacterial TMDL Task Force, which I was a part of. All right, first thing I want to just point out is that source tracking projects tend to take a lot of different partners, collaborators for those projects. I want to recognize the folks from my laboratory that participated in some of the BSD projects in the past and currently. As well, if you look at some of the collaborators, we have organizations like the Texas Water Resources Institute, Parsons Consulting, James Merchant and Associates, and also a consulting firm, and then other stakeholders like Texas Farm Bureau and the City of Waco for one of the projects that we did, Brazos River Authority, as well as funding by state regulatory agencies like the Texas Commission on Environmental Quality and the State Soil and Water Conservation Board, with money also coming from the Environmental Protection Agency. All right, solving water quality problems. This is how we got to where we are right now, okay? Clean Water Act, of course, and in the Texas water quality criteria, E. coli being the organism for all the inland fresh water sources, with a long-term geometric mean not to exceed 126 CFU per 100 ml for E. coli. There used to be a maximum sample density, but the state's trying to do away with that portion of the regulations. Currently, on the 2008 impaired water body list for the state of Texas, there are 274 water bodies identified as impaired by elevated bacterial counts. That is fecal coliform historically, but also E. coli counts as well. So plenty of places to do studies. Okay, fecal pollution, what do we track? Okay, we've got chemicals, chemical methodology like caffeine, brightening agents. These are optical brighteners which are used in laundry detergent, and those can be very useful, and some source tracking people have used those. Fecal sterols are another group of compounds that have also been used. Those are, of course, as you just hear about them, they're more human related. And so if you're looking at water that's impacted by animal fecal pollution sources, you might need to use some other methodology. Viruses, which are very specific to their hosts, you'll hear a little bit more about that in another talk for this special topic session. Those can be very useful, but sometimes they're expensive to analyze for. It's one of the limitations, and they're not always present in your samples at the same levels. Of course, you find fecal bacterial indicators. Parasites like Cryptosporidium. We do a lot of work with Cryptosporidium and related parasite Giardia in my laboratory. There are ways of also doing molecular source tracking to hosts based on those organisms, but cost is still an issue, and abundance in fecal pollution is also an issue. So we're talking about bacteria now, and two groups in particular, the Bacteroidales or Bacteroidetes, if you want to be more purist as a taxonomist. E. coli, of course, the basis for the water quality regulations in Texas right now. Enterococcus have also been a focus of bacterial source tracking approaches. So there are E. coli in the water, but where do they come from? It's very important to know because we were trying to develop and implement best management practices with DFPs. You won't hear me say the nasty acronym TMDL. For those of you that know what that is, let's just not even think about it right now. We can talk about that later. Okay. So bacterial source tracking used as a tool. And think here, well, tracking fecal pollution sources using E. coli. What's that based on? It's based on the premise that different animal guts or environments within that host animal are different, so we have different adaptations and selection of different E. coli strains within those animal populations. Those adaptations result in genetic differences and phenotypic differences that we can detect using BST analytical techniques. So we have lots of different critters here. We've got pets, livestock. Feral hogs, a big issue in Texas. Doing something that this fellow reading his newspaper. We have some more exotic animals over here and laugh about that. We're like, oh, you got lots of rhinos running around in that watershed, right? Well, actually, one of the studies that we did in the San Antonio area had a river impacted by a discharge from the San Antonio Zoo. So, yes, we had to look for rhinos. Okay, so the BST using laboratory tests to determine if E. coli or other fecal bacteria or other organisms, if we want to call it microbial source tracking, came from animal or human feces, animal or human fecal pollution sources. So that's the whole objective there. Many of the BST methods that have been used in the past are library dependent. They're library dependent techniques, which means that we need to have a database of reference bacteria. In particular, I'm going to talk about E. coli that have been isolated from known animal or human fecal sources. So we have to go and isolate E. coli bacteria from animal and fecal samples, animal and human fecal samples, characterize them with the BST methods, and then include them in a library. So when we get E. coli isolates from water samples, we can type them and try and match them up to what we have in our library for known source ID. Local watershed libraries, local libraries, those libraries built on E. coli isolates from fecal specimens or sewage samples or septic samples in the particular watershed that you're using are typically considered the most useful. And one would expect that because you're narrowing down the whole diversity of E. coli that you might find around the whole world to those that are typically occurring in pollution sources in the particular water body that you're studying. Okay. The problem with that and library methods is that when you go to another watershed, you have to build another local library. That can get very costly and take a lot of time. Okay, so that's one of the major disadvantages of the library-based methods. Well, welcome to the poop show. This is a real sign too, by the way. It came from the San Diego Animal Park. So they have a poop show for kids, but for us, it's a poop show for myself and my staff, unfortunately, because building a library means that we have to analyze those fecal specimens to get the E. coli isolates or extract bacterial DNA from them, like we'll also describe for some of our library independent methods. So working with fecal samples is really stinky. All right. Just to give you an idea about a couple of our projects, these were finished a couple years ago. One of them was in the Waco, Texas area. We refer to it as the Soil Board or the Waco Project. And this just gives you an idea of the magnitude of some of the studies. 3,061 E. coli isolates from 765 different source samples, that is, different fecal specimens. Okay, so that's a lot of fecal samples to work with. 634 E. coli from 415 different water samples. It ended up being, we had some false positives for E. coli in there. Some were excluded because of their inability to be typed by one of the methods that we're using. So we had 555 isolates from 412 samples. Same kind of study size here for the San Antonio project. 3,000 E. coli from 759 source samples. See, we ended up over here. We whittled that one down considerably. That's because this library was created first, and we did some elimination of clonal isolates and things that we'll talk about briefly. But a lot of water samples, lots of isolates, lots of E. coli to work with. One of the important things, and you heard some talks earlier today about microbial ecology and selection of organisms in the environment. It can also happen when you talk about selecting cultural organisms from environmental samples or from fecal samples for that matter. In the case of E. coli, some of the other studies that have been done, bacterial source tracking studies, have used clinical type media for the isolation of E. coli. In these studies, we've used US EPA